NMIT presents Lecture 9 on the topic of translocation in the phloem. This is part of the plant physiology subject which is offered as a component of both the agricultural and viticultural degree programs at NMIT. Please refer to the recommended reading from Tays and Zeiger, Chapter 10 for complementary reading. Please ensure that you have watched Topic 5 on the topic of plant solutes before continuing with this lecture. My name is Dr. Nikki Cooley. This topic will be separated into two lectures. Part 1 will look at phloem function and structure, while Part 2 will look at loading and unloading in the phloem, and allocation and partitioning. Please watch the following YouTube video. This gives an overview of solute transport and will bring you up to date. Please also ensure that you have covered Topic 5 of this subject before completing this lecture series. For plants, the survival on land poses serious challenges. They need to acquire and retain water. As plants increase in size, leaves and roots increasingly separated from one another. Thus, systems evolve for long-distance transport of products of assimilates and absorption. The xylem transports water and minerals from the roots to the aerial proportions of the plant, while the phloem has a different function. It transports products of photosynthesis from mature leaves to areas of growth and storage, including the roots. Phloem also transmits signals between sources and sinks, and redistributes water and various compounds. There are two transport pathways, the phloem and the xylem, and then they extend throughout the plant body. The phloem is generally found in the outer side of both the primary and secondary vascular tissues. The figure on the side illustrates both xylem and phloem structure. Cells of the phloem are called the sieve elements. These include the sieve tube elements in the angiosperms and the sieve cells of the geosperms. In addition, the phloem tissue contains companion cells and parenchyma cells. These store and release food molecules. In some cases, phloem tissue contains fibers and spheroids. They protect and strengthen the tissue. And lacifers, which are latex-containing cells. The small veins of leaves and the primary vascular bundles of stems are often surrounded by bundle sheath cells, as can be seen in the illustration on the slide, figure 10.1 from their recommended text. One or more cell layers are involved in C4 photosynthesis. Mature sieve elements are unique among living cells. They lose their nuclei and toneoplasts, vacuole membranes. Their microfilaments and microtubules, Golgi and ribosomes are also generally absent. The plasma membrane is retained in addition to modified mitochondria, plastids and smooth endoplasmic reticulum. Different to tracharine elements of the xylem, which lack in the plasma membrane, they have ligafied secondary cell walls and are dead at maturity. The figure on the slide illustrates the structure of a mature sieve tube element. I will draw your attention to the sieve plates and how they look, containing sieve plate pores and lateral sieve areas. Also, in Part B of this figure, you will see sieve tube elements along with companion cells. Sieve elements have characteristic sieve areas in their cell walls where pores interconnect the connecting cells. Sieve plates have larger pores and are found on the end of walls of the sieve tube elements where they are joined together to form a sieve tube. Sieve plate pores are open channels allowing transport between cells. Sieve element sap is rich in sugars and other molecules, representing an energy investment by the plant. Therefore, loss of sap due to damage must be prevented. Short-term sealing mechanisms, or sap proteins, slime. And there are long-term mechanisms to prevent the loss of this material, closing sieve plate pores with callose. Callose deposition is induced in rice plants attacked by phloem-feeding insects such as a brown plant hopper or aphid. This occurs in both resistant and susceptible rice plants. However, in susceptible rice plants, feeding by insects also activates genes for the callose hydrolyzing enzyme. This unplugs the pores and allows continuous feeding, and results in decreased sucrose and starch levels in the leaf. Each sieve element is associated with one or more companion cells. Division of a single mother cell forms a sieve tube element and the companion cells. The function in the transport of photosynthates from producing cells in mature cells into the sieve tubes elements in the minor veins of the leaf. They also take over some of the critical metabolic functions that are reduced or lost in the sieve tube cells. For example, they supply ATP, the energy compound the plant uses. These companion cells come in at least 3 types, around the minor veins. The first type, the original companion cell. This is with a cell wall with a smooth inner surface. The second type are called transfer cells. They develop finger-like ingrowths of the cell wall primary on the walls that face away from the sieve element. They greatly increase the surface area of the plasma membrane. They increase the potential for solute transfer, few plastomata, and they are suited to apoplasmic transport. The third type is the intermediary cells. They have numerous plasmodesmata, connections to bundle sheath cells, also have numerous small vacuoles poorly developed by thylakoids. These are well suited to symplastic transport of sugars from mesophyll to sieve cells. The image on the slide shows 3 visual representations of companion cells. Translocation is the term used for the movement of minerals and substances in the phloem. Translocation is not defined by gravity. Rather, it moves from areas of supply, or what we refer to as sources, to areas of metabolized or stored compounds called sinks. Sources include exporting organs, mature leaves that can produce photosynthetic substances in excess of their own needs. A storage organ exporting its stored sugars is also called a source. For example, the storage root of wild beet is a sink during the growing season. Then during the second season it becomes a source, remobilizing sugars to produce a new shoot. Sinks include any non-photosynthetic organ and organs that do not produce enough photosynthates. Sink tissues include roots, tubers, developing fruits, and immature leaves. The overall pattern of transport is more complex than simply source to sink movement. It depends also on the proximity, development, vascular connections, and modification of translocational pathways. Certain sources supply specific sinks. Please watch the following video on phloem-source-sink relationships. This will illustrate a visualization of the process that we are going to describe in the lecture today. Water is the most abundant substance in the phloem. The phloem also contains carbohydrates, amino acids, hormones, some inorganic ions, RNases, and proteins, and some secondary compounds involved in defense and protection. Carbohydrates are the most significant, with sucrose concentrations ranging from 0.3 to 0.9 molar. The table on the screen, table 10.1, gives a composition of phloem sap from bean plants. It is commonly accepted that reducing sugars such as sucrose, mannose and fructose are not translocated in the phloem, possibly too reactive in this form. Non-reducing sugars are commonly translocated. Also the sugar alcohols, mannitol and sorbitol. The figure on the screen shows the structure of some of these compounds. It is quite challenging to tap the phloem sap due to the high turgor pressure and wound reactions which seal off damaged elements. Aphids have evolved methods such as using a stylet as a natural syringe, where they are able to both overcome the protective mechanism and access the contents of the phloem. Other solutes in the phloem include nitrogen, which is in the form of amino acids, glutamate and aspartate. Plant hormones, which include auxin, the family of gibberellins, cytokinins and ascorbic acid or ABA. Low levels of proteins, mostly related to stress and defense mechanisms. There are low levels of RNA, mostly in the form of messenger RNA, which are small regulatory RNA compounds. Some inorganic solutes can be found, such as potassium, magnesium, phosphate and chloride. Nitrogen, calcium, sulfur and iron immobile in the phloem. A model called the pressure flow model has been developed to explain the phloem translocation. The model poses that translocation occurs as a flow of solution, that is, the mass flow over the bulk flow, and it is driven by pressure gradient between the source and the sink. Diffusion is too slow, i.e. if translocation velocities average 1 meter an hour, the rate of diffusion would be 1 meter per 32 years, far too long. The pressure flow model was proposed in the 1930s. Phloem loading is the movement of photosynthates into the sieve elements. Phloem unloading is the movement of photosynthates from the sieve elements to the sink cells. Under this model, 3 different mechanisms exist to generate high concentrations of sugars in the sieve elements to the source. 1. Photosynthetic metabolism in the mesophyll cells. 2. Conversion of photoassimilates to transport sugars in intermediate cells. This is called polymer trapping. And 3. Active membrane transport. The slide shows a visual representation of the pressure flow model. You will note the source cell, the companion cell and the phloem sieve elements. You can follow the route that a sugar will take from the source cell, illustrated here as sucrose in red spheres, and how it is actively loaded into the phloem sieve elements. You will see the partial pressures involved in this process. If you then follow the solute down to its final destination, the sink, the sugars are unloaded and are then transported into the sink cell where they can be used or stored. The pressure flow model states if no cross walls were present, i.e. if the pathway was a single compartment, the different pressures at source and sink would equibrate. Sieve plates present a sequence of resistances to the moving phloem sap. This resistance results in the generation and maintenance of the pressure gradients between source and sink. The second component to note is that mass flow permits the flow from a source organ with a lower water potential to a sink organ with a higher water potential. Is this a violation of thermodynamics? No, as it is mass flow, not osmosis, which is driven by the water potential gradient. So in summary, the pressure flow model illustrates movement in the translocation pathway is driven by transport of solutes and water into the source sieve elements and out of the sink sieve elements. The passive pressure-driven long-distance transport in the phloem depends on phloem loading and unloading. These mechanisms are responsible for setting up the pressure gradient.